Review sheet for second lecture exam. So, it's going to be held on Wednesday, this coming Wednesday, October 16th at 10 o'clock in our classroom, SB 110. It'll be like the first exam objective. So you're going to have multiple choice, true, false, matching, identify figures, but you're also going to have a genetic code question. So off and away we go. The first PowerPoint is on enzymes and metabolism. So what I would do is look at your class notes. I would look at the PowerPoints, and then I would look at the review sheets. And what the review sheet is trying to do is to highlight everything. So repetition is the mother of learning. Do all of that. So let's begin with enzymes and metabolism. So it says enzymes act as a catalyst, a biological catalyst, because they're in the body. They speed up the reactions. Now, the lowering of the energy of activation you don't need to know, but you do need to know that they are enzymes or proteins act as biological catalysts that speed up reactions in cells. Now, the enzyme does have an active site, which is lock and key specific for the substrate which bind. So if you have triangles, then you should have receptors that would be able to specifically bind triangles and not circles or squares. Then once the reaction is complete, the new products are released and the enzymes can be used again. And I think that's an important point. It's not like you use up an enzyme. It is a catalyst, it makes things go fast, but you don't consume it. So if you have molecule AB, that they are bound to each other and they bind to the active site, they're close enough to be able to break that bond very quickly. So the enzymes act as a place where reactions can occur fast. But the enzyme itself is not harmed and you can reuse it. Now the next part has to do with metabolic enzymes. The metabolic pathways that are in the cell. And we are going to cover aerobic respiration, for example. What you'll find out later in your sophomore, junior, and senior year is the name of all those enzymes in the pathway. But metabolic pathways can be regulated, so they can be shut off, they can be shut on, or they can be turned on. So you want to do that quickly as well. So the first enzyme in a metabolic pathway, which could have 10, 15 steps to it, is the allosteric enzyme. Allosteric means other site. So you have your active site and you have your allosteric site. You have your active site and you have another site that'll bind the end product of the pathway to turn the enzyme off. So you'll have that allosteric site or other site which can bind the end product of the pathway. And if the end product is no longer being used, then it'll bind to the allosteric site on the first enzyme and shut down the entire pathway. This is called negative feedback. A negative is kind of a bad, well, it's not a terrific word. The feedback is negative because it's shutting something down. So if you have a pathway and you're using an end product, great. If all of a sudden you are no longer using that end product, that's the negative feedback. The end product will feed back to the allosteric site on the first enzyme, bind, and then shut down that first enzyme. How? So the binding of the unused end product to the allosteric site causes a shape change. We call that a conformational change in the scientific medical lingo. Now, this shape change caused by the end product binding to the allosteric site changes the shape of the active site. So the substrates can no longer bind. This effectively shuts down the entire metabolic pathway thus assuring that end products no longer need by the cell are no longer made. But it is reversible, so if a cell starts to need the end product again, it can remove it from the allosteric site, and the active site is restored and can now bind the substrates, activating the entire metabolic pathway once again. So, since each step of this metabolic reaction depends on a product that can specifically bind, if you take that first enzyme and the end product has the allosteric enzyme or allosteric site binds to it, then it's going to change that active site and that enzyme will no longer work. And then if that enzyme doesn't work, then you don't make the product for enzyme 2 and so forth and so on. So it's a very sophisticated way, very easy way of shutting down a pathway, but it's reversible. So if you need the end product again, you pull it off that allosteric site on the first enzyme and away you go. So that is enzymes, metabolism, and allosteric enzymes negative feedback. So the next slide, I just suddenly went back to the first slide. Next slide is about the types of metabolism. And you have two types. Catabolism means breakdown. Anabolism means synthesis. And again, in your sophomore and junior year, you'll be taking classes where they will go into specific catabolic and anabolic reactions besides the ones that we're going to do. The catabolic reactions that we're going to do on this exam is going to be aerobic and anaerobic respiration. The anabolic or anabolic reactions that we're going to do on this exam is DNA synthesis, RNA transcription, protein translation. Okay, so in catabolism, you have the degradation of a molecule where chemical bonds are broken and energy is released. That energy can now be used and trapped in the form of ATP. In addition, the breakdown of molecules provide the cell with small building blocks. So think Lego blocks. You have a Lego tower, you want to make a Lego bridge. First thing you do is you take apart the Lego tower. In the world of catabolism, doing that, breaking those Legos would release energy trapped in ATP, and it would always give you at the end a whole pile of different types of Lego blocks in which you can now build your Lego bridge. Now, the anabolism or synthesis always comes with two demands. It comes with the demand of energy in the form of ATP, and it comes in demand of building blocks. Because if it has energy and it has building blocks, it can make whatever it needs. So the building blocks and the ATP that you got from catabolism can now be used for anabolism or synthesis by the cell. So the examples, we're going to do aerobic respiration as an example of catabolism, fermentation, anaerobic respiration, another example, and synthesis or anabolic reactions are the DNA replication and RNA transcription and protein translation. And in those particular processes, you don't see, or we don't really track how much ATP goes in or building blocks go in, but they do. So, having said that, let's whip right along to cell respiration, aka aerobic respiration, and fermentation, aka anaerobic respiration. So what you have is aerobic, we're going to do first. It's the more complicated of the two. Here we have glucose and oxygen entering a cell. It's going to be broken down by the metabolic pathway called aerobic respiration to form 38 ATP, carbon dioxide, and water as waste products. It takes place in 3 stages: glycolysis, which takes place in the cytoplasm; Krebs cycle, which takes place in the middle or matrix of the mitochondria; and the electron transport chain, which is located on the inner folded membrane of the mitochondria. All right, this is kind of a slide that incorporates a lot of stuff, but you also have your metabolic worksheet, which we went over in class. And we also went over in class that overview of the 3 stages. Lots of you took pictures. You did a fantastic job on the hybrid. Look at those. Memorize them so that you can apply your memorized knowledge of it and be able to solve problems. So glycolysis, the 6-carbon glucose is broken down into 3-carbon pyruvic acids or pyruvate. And it makes, in doing that, 2 ATP. We call that substrate level phosphorylation. Why? Because you took a molecule, a substrate like glucose, you broke it in half, and that released energy when you broke that bond. Because you're breaking the bond of a molecule, not an electron, but in a molecule, that's called substrate level phosphorylation. Then each one of those pyruvate or pyruvic acids is transformed into acetyl-CoA when carbon dioxide is removed. And the other thing that happens is there's a hydrogen pair that is also split off and goes by the taxi of NAD to the mitochondria. And I managed to forget, I didn't forget it, but it's right here. You make an ATP and you make 2 hydrogen pairs. Okay, so you break glucose, C6, into 2,3-carbon pyruvate. You make 2 ATP and you break off 2 hydrogen pairs. When you take the pyruvate or pyruvic acid and get rid of the CO2 and get rid of another hydrogen pair and get CoA to be a taxi for that 2-carbon acetyl group, then that is that transition between the cytoplasm and the mitochondria. So the mitochondria is going to first begin the Krebs cycle. And the acetyl-CoA goes into the matrix of the mitochondria. The 2-carbon joins 4-carbon to form a 6-carbon molecule. The 6-carbon releases a CO2 and a hydrogen pair to form a 5-carbon intermediate. This, in turn, is transformed into a 5-carbon molecule, and another 2 hydrogen is formed. It's cut off by NAD, taxied to the electron transport chain. The 5-carbon becomes 4-carbon. You release a CO2. You take 2 hydrogens and taxi them by NAD, 2 hydrogens taxied by FAD, and you make 1 ATP by substrate level phosphorylation. Now we're back to 4-carbon, and we can begin a new cycle when the second acetyl-CoA from the second pyruvate or pyruvic acid enters the mitochondria. So that is just a reminder that you always have that other pyruvate or pyruvic acid waiting in the wings for the first one to go around so it can then do a second turn of the Krebs cycle. Okay, then we have the electron transport chain, also called oxidative phosphorylation. Cytochromes on the inner membrane of the mitochondria begin to process all of those hydrogen pairs one at a time. They line up like people line up to get on a Ferris wheel, that type of thing. Each hydrogen pair is cleaved, and high energy electrons begin to travel down the electron transport chain where the cytochromes bind and release electrons and trap the released energy into ATP. Now, there are 3 ATP for every pair of hydrogen electrons that travel down the cytochrome stairs, which they're dropped off by the taxi of NAD. So think about that hot potato game where the cytochromes toss the electrons and accept them, toss them to the second cytochrome, accept it, toss it to the third cytochrome. Each time they toss the electrons, they release a little energy, trap it in ATP, So going down from cytochrome 1 to 2 to 3, you make 3 ATP. The only exception is FAD. It's going to be a taxi that will only create 2 ATP for the hydrogen that it drops off. All right, so the ATP is formed by the electron transport chain. It's called oxidative phosphorylation because oxygen, that's the oxidative, oxygen is going to be at the end. The oxygen will reunite the hydrogen electron, the hydrogen proton, and along with the oxygen, you make the end product of aerobic respiration, which is H2O, or water. Okay, so the next slide is showing you where you get the 38 ATP from one molecule of glucose. And I want you to understand this. So if I ask you different questions about it, then you would be able to answer. Very similar to the questions on the metabolic worksheet. So we know from substrate level phosphorylation, we get 2 ATP from glycolysis and 2 from Krebs, because it goes around twice, to give you a total of 4. Oxidative phosphorylation by NAD, so glycolysis produces 2 hydrogen pairs. That's the 2 right there. Each one of them will produce 3 ATP, so that gives you a total of 6. Well, you get 2 from pyruvate to acetyl-CoA because 1 pyruvic acid gives you 1 pyruvic acid gives you 2. So now you've got, again, 2 of your hydrogen pairs times 3 ATP gives you another 6 ATP. 1 turn of the Krebs cycle produces 3 hydrogen pairs taxied by NAD for each pyruvate broken down. So that's 3 times 3 is 9. You have 1 NAD that is going to drop off a hydrogen pair, but you're only going to get 2 ATP for that. And that has to do with the fact that the FAD is just slightly different. And so that is going to give you 2 ATP. But the Krebs cycle turns again with the second pyruvic acid. So now you've got another 9. So each glucose yields 4 by substrate level phosphorylation, and by oxidative phosphorylation, you get 18, 4, and 6. Okay, I'm sorry, 18 and 4 are 22. You get another 6 from glycolysis and another 6 from pyruvic acid and acetyl-CoA. So now you've got 18 plus 4 plus 6 plus 6 is 34. And then last but not least, we're going to add another 4 to the 34 to give us 38 from the 4 ATP made by sub-level phosphorylation. Yay! Next slide. Now we're off to cell cycle. Half the cells in your body replace themselves all the time. So they go through this cell cycle where they're going to end up doing a lot of metabolism, replicating the DNA, getting ready for cell division or mitosis. The old-time terms of cell cycle is mitosis and interphase. We know, however, that interphase is actually composed of a G1 phase, an S phase, and a G2 phase. Then you can do mitosis. So body cells replace themselves. They undergo a process called cell cycle. So a typical epithelial cell takes about 24 hours to go through a cell cycle and to replicate, make 2 new cells. So I do want you to know the emphasis of where you spend the most time of the 24 hours in cell cycle. The first stage is cell division of mitosis, and we'll cover that in a couple more slides. That takes about an hour. Then the cell enters the first gap phase, and about 20 of the 24 hours, in other words, the majority of the time, is spent in G1. During G1, the cell synthesizes proteins, expresses itself, does metabolism, blah, blah. But then it knows it's going to have to divide pretty soon. So it swings into the S or synthesis phase where it does DNA replication. Takes about an hour. Once you've replicated the DNA, you are going to go into the second gap phase, or G2, and it takes about 2 hours. And during that second gap phase, you finish up any leftover projects that need to be done before the cell swings into mitosis. However, there are some cells in the body that have left cell cycle permanently. They did their last cell cycle when you were a fetus, and then you were born. Your brain cells have left cell cycle, your heart cells have left cell cycle, your kidney cells have left cell cycle, but they are alive, they're well, they express, they do fantastic things in the body. But if you lose them due to injury or disease, they're gone. You can't replace them. They will not go back into cell cycle. They're just gone. And in the case, let's say, of the kidney, if you have a kidney disease and after you get into recovery, you've lost 20% of renal function, you've lost 20% of renal function. You don't get it back. So you've got 80%, but you don't have 100. So the G1 phase is called, or G0 phase, is for all the cells in the body that are so highly specialized, they've left cell cycle and they cannot go back. All right, DNA replication and DNA condensation. So actually, let me do replication first because it comes first. Before you divide a cell, you have to make a copy of the chromosome so that one cell is going to get a complete set of DNA instructions and the other cell is going to get a complete set of DNA instructions. The process of making a copy is called DNA replication. We know that DNA is a double helix. It's composed of nucleotide monomers. We know that a phosphate is bound to a sugar, which is bound to a nitrogenous base. And DNA is going to have 4 different nitrogenous bases. Just before cell division, during the S phase of the cell cycle, the DNA double helix unwinds and it breaks the hydrogen bonds. Remember, they're the weak hydrogen bonds that hold the nucleotides, specifically the nitrogenous bases, together. All right, so we have the 4 bases as adenine, guanine, cytosine, and thymine. The base pairing rule states that A binds to T, that makes that weak hydrogen bond, and G binds with C, and makes that weak hydrogen bonds in the DNA double helix. The sides of the helix are the sugar phosphates, and the middle are the rungs of the ladder that are the A and T and G and C pairing. The A and the T and the G and C are actually held together by weak hydrogen bonds. I just couldn't figure out how to do this with my keyboard. And once the DNA unwinds, an enzyme called DNA polymerase links the new nucleotides together. So when it unwinds, you've got a stockpile of individual nucleotides, and you just bring them in. A's will bind with the T's, G's with the C's, and so forth and so on. Once you've got all of those, the polymerase takes that new strand and makes it hang as a whole. And then you form your 2 new strands off of your original template, and then you are able to do mitosis and get those chromosomes to each of those 2 new cells, so the cells are exactly like the original. This type of replication is called semi-conservative, since one strand is the original, or the old strand, or the conserved strand, and the other one is the newly synthesized strand. Okay, that's replication. Now we got to coil the sucker and condense the sucker to make chromosomes so you can see them in mitosis. That's called condensation. So just before mitosis, the DNA condenses into chromosomes. The first condensation is DNA winding around histone proteins. The second is when the histone proteins with the DNA wind around each other to form a nucleosome. The nucleosomes coil into chromatin strands, and then these chromatin strands will condense into chromosomes. We have 46 in each cell in the body, 23 from mom, 23 from dad. So you are a diploid organism that was made from a unique combination of 43 chromosomes from mom's egg, 23 chromosomes uniquely blended in dad's sperm. And then when they came together for fertilization, you got your 46. 9 months later, you got your baby. All right, mitosis, cell division, finally. So what we have is we have cell division in 4 stages: prophase, metaphase, anaphase, telophase. And in that last step, the cytoplasm is dividing as well, and that's called cytokinesis. Know what these stages look like. We went over it in lab. You looked at onion root tips. You have it in your PowerPoint. So make sure that you know what each stage looks like for identification. So we have prophase, metaphase, anaphase, telophase. Cytokinesis takes place during telophase. A body cell is composed of 23 maternal and 23 paternal chromosomes for a total of 46. Okay, so prophase. The chromosomes condense. They are held together by a protein centromere in the middle. The nuclear membrane disappears. The centrioles divide, and they migrate to opposite poles. The centrioles spin out spindle fibers that attach to the centromere, that button-like protein of the chromosomes, not, but the centromeres are proteins, but it's going to be in the middle of the chromosome. So that was a very awkward sentence. Metaphase, the chromosomes line in the middle of the cell. Anaphase, the centromere splits. So the spindle fibers are going to start to pull those chromosomes to opposite poles. And then in telophase, they finally arrive at opposite poles. And the chromosomes then, once they've arrived, the nuclear membrane reforms. During this period of time, the cytoplasm is also dividing in half, splitting the cell in half, and that process is called cytokinesis, division of the cytoplasm. And the other thing that is in here that should be in here is that once you've got that nucleus reform, you're going to decondense the DNA. It's going to return to that spaghetti-like mess in the nucleus. But that is something that I forgot to include. So telophase, it gets to the opposite poles, you decondense the chromosomes, you reform the nucleus, and then cytokinesis also occurs, and you've got yourself 2 new cells. They are exactly alike. It's kind of like cloning, and then each one of them have a full set of chromosomes and can do whatever the cell dictates. So that is mitosis. The 20 of the 24 hours that a cell stays in cell cycle is going to be a time of transcribing messenger RNAs from the DNA template, taking that messenger RNA out into the cytoplasm, where you can bring to that messenger RNA ribosomes, amino acids, transfer RNAs, to then synthesize a 3-dimensional protein made of amino acids. So you're going to have information flow from DNA to messenger RNA to protein. And this is all happening during that G1 phase. So let's talk about transcription of message. when a cell needs to make a protein, the information or blueprint is stored in the nuclear DNA. A copy has to be made. The copy is a closely related molecule of ribonucleic acid. How does it differ? It differs in that it is single-stranded and it uses the nucleotide of uracil instead of thymine. It's important to make a closely related molecule but not have it be DNA because the RNA is single-stranded. It'll get through the pore and get into the cytoplasm. And since it's a copy of the blueprint, the copy works just fine out of the cytoplasm where you're going to build your protein. So the area that encodes for the blueprint unwinds the RNA polymerase, synthesizes one strand of the DNA. The single-stranded messenger RNA leaves the nucleus, goes into the cytoplasm. The DNA is then going to rewind, and the RNA transcription is complete. Now, in the cytoplasm is where you're going to build your protein. You're going to build your three-dimensional protein made of amino acids. The process is called protein translation. It can also be called protein synthesis. And so in protein synthesis, you have your messenger RNA, you have your ribosomal subunits, you have your transfer RNA, and you have your amino acids. The messenger RNA is embraced by the ribosomal subunits when it comes into the cytoplasm. The transfer RNA, or tRNA, is a recognition molecule. It can recognize the RNA codons, and it can bind as anticodons. It also recognizes and binds the proper amino acid to load, so that the transfer RNA molecule is going to have one end amino acid, another end a three-letter RNA, where you're going to bind A with U and G with C. So they're complementary to the messenger RNA. See, what we found out is that you only have 4 nucleotides for DNA, but you have 20 amino acids. So what they figured out is that to make one amino acid, you use 3 letters of the nucleotide, and they call the 3 letters codons. So when the transfer RNA comes in and binds to the messenger RNA codon, they had to call it something else. So they called it an anticodon. Terrible word, but I mean, because it's not anti-anything. It's just the complementary sequence that can bind to the messenger RNA codon, but it's called anticodon. There you go. Now, the messenger RNA is read left to right, 3 at a time. And each of those 3 is called a codon because it's going to represent one amino acid. So the ribosome is going to slowly move down the messenger RNA, and the amino acids are going to slowly form peptide bonds as the amino acids are added on and added on and added on until you get to a stop command, and then the protein is complete. The ribosomes will disengage from the message, and the message will release the newly formed protein. So during class, we worked on a worksheet called the genetic code worksheet. You will have a copy of the genetic code worksheet in front of you. So do not memorize it, but know how to read it. So it says the circular genetic code contains the first nucleotide of the triplet in the middle, the second nucleotide in the second part of the genetic code, and the third letter is going to be on the third circle. So it says, be sure you understand the genetic code worksheet since it'll appear on the exam along with a copy of the genetic code, and look at your worksheet to help you. So here's one. This is what you will be having right in front of your face. So if I have a codon that is AUG, it means that the A is going to be in the center. That's your first letter of your codon. And that means that the A is going to be in this portion and this portion of the genetic code. The second letter is A, so it's the second circle. And the third letter is A, which is your third circle. I'm sorry, so it's AUG. Sorry about that. A is the first one, U is the second one, and then G is the third one. So A, U, and G. And it stands for, these are three-letter abbreviations of amino acids. So MET, M-E-T, is an abbreviation for the amino acid of methionine. Let's do AAA since I mentioned it. So AAA, the codon, is going to represent the amino acid of, right? A is the first letter here, A is the second letter, and A is the third letter, and it's the amino acid that is abbreviated LYS. And when you take biochemistry, you're going to know that that corresponds to the amino acid of lysine. So each one of these in that fourth circle is an abbreviation of the 20 different amino acids. Just for your edification, gly is glycine. Don't need to know this for the exam, just for the three letters. But I know you're dying of curiosity. Phe is phenylalanine, Lu is leucine, Ser is serine, Ty is tyrosine. That's a stop command. So remember that your protein is like words in a sentence where the sentence is composed of three-letter words like run, dog, run. But at the end of run, dog, run, you have a period, and it tells you that you're at the end of the sentence. So the stop commands work in the same way. They're like a period at the end of a sentence, so you know when you have gotten to the end of the amino acid chain of the protein. Cis stands for cysteine, another stop command. Trip for tryptophan, Lu for leucine, Pro for proline, His for histidine, Glyn for glutamine, Arg for arginine, IL for isoleucine, and MET for methionine, 3 for threonine, ASN for asparagine, Lys for lysine, Ser for serine, Arg for arginine, Val for valine, A for alanine, Asp for asparagine, Glu for glutamine, Gly for glycine. You can tell I'm a biochemist. So this is the genetic code that you'll have. You'll know how to do that first, second, and third codon and the three-letter abbreviation for the amino acids that make up the protein. And that's it. So have fun studying on the weekend. Monday is Indigenous People Day. We have the day off. No lab next week. You have a lab midterm. It's open book, individual effort. You have all the Moodle labs in your Moodle course. You have your lab notes and the results you got. So work on that. It's due a week from Indigenous People Day, so it's due the 21st. But this second in-class paper exam is going to be on Wednesday, the 16th. So have fun, and I will see you soon. Take care.